Welcome back to our fourth and final lecture in the reproductive system and the very last lecture of the class. Let's go ahead and begin. So last time we were talking about the structures that will be responsible for delivering the sperm from the testes out through the man's body. So this image clearly shows the seminiferous tubules. So here's where sperm are created by the process of spermatogenesis. As they mature, they move towards the lumen of the seminiferous tubule, which will then carry them out the retrotestis, out the efferent ductal to the head, body, and tail of the epididymis, then flow through the ductus deferens. It brings us to this point right here, the ductus deferens. The ductus deferens will eventually lead to the ejaculatory duct, which is found inside the prostate or prostate gland. And from there, it will connect to the urethra. Right, so I've already talked about these structures, retrotestis, seminiferous tubules to the efferent ductals to the epididymis, which has three structures. Right, one of the points, the purposes and functions of the epididymis we talked about last time is it's going to store sperm until they're needed and actually serves as the final maturation point for sperm. So sperm are not fully mature when they leave the seminiferous tubule, when they actually leave the testis. Final maturation will take place in the epididymis. And one little factoid is sperm actually don't start swimming. They don't start expending their energy until the actual ejaculation, by the time they're actually called upon to function. Otherwise, they're waiting in a dormant state. Okay, so this is an image from the posterior showing all the structures that we've just talked about. So in here, we have a lobule filled with seminiferous tubules. We're at kind of a low magnification, so we can't see the retrotestis or the efferent ductals, but we can see the head, body, and tail of the epididymis leading to the ductus deferens. Ductus deferens is going to go up and over the ureter. So in this area, the ductus deferens will widen into a structure called the ampulla, and then it will meet with the duct of the seminal vesicle. So the seminal vesicle is a gland that produces some fluid that is a component of semen. So I should point out here, there's a difference between sperm and semen. Sperm are little swimmy guys carrying half the genetic information, right? Semen is actually a product of three glands, three organs: the testes, the seminiferous vesicle, the seminal vesicle, and the prostate itself. So you need three different structures, all adding material before you actually get semen. So there's a difference between the two. So here at the end of the ductus deferens, right, you have a structure called the ampulla. It just gets a little bit larger and wider. Here it meets the duct of the seminal vesicle to form, those two tubes come together to form the ejaculatory duct, which is inside the prostate gland, where the prostate gland is immediately inferior to, it's underneath the bladder. So the ejaculatory duct will open up into the prostatic urethra. Now we're sharing reproductive and urinary structures. Prostatic urethra, the membranous urethra, and then the spongy urethra. And the reason this is called the spongy urethra is this is called the spongy body, or corpora spongiosum. Right out here is a different structure called the corpora cavernosa. So there are actually two corpora cavernosa, one corpora spongiosa. So we have three erectile structures or erectile bodies. The way that these structures work is the parasympathetic nervous system will allow blood in, so you're going to get vasodilation on the arteries, and the exit point, the veins, will be closed off. So blood will flow into this structure, but it will not be allowed to flow out. So these tissues basically swell with blood, and you have hydrostatic pressure generating an erection. This is the same basic principle that soft-bodied plants use, right? So your, house plants and things like that, you water them and they kind of stand up, and if you don't give them enough water, they're kind of drop over. Same basic principle here in the penis. Let's see here. Other structures. We have these bulbourethral glands. These are directly homologous to the vestibular glands, also known as the Bartholin glands in female anatomy. And they serve the same purpose. They're going to produce a liquid, a thin mucosal material that also helps serve as a lubricant during intercourse. Let's see, anything that I have missed at this point? Nope, think I've talked about pretty much all of it. Oh, here we have a cross-section of the ductus deferens. You can see we have a very thick mucosa, very thick muscularis. So this is a fibromuscular tube, right? The smooth muscle is going to help propel sperm where they need to be, right? Mucosa, pseudostratified, ciliated epithelium. It kind of makes sense because like we had in the uterine tube, right? Part of the purpose of this, right? It's going to be ciliated to help get sperm where they need to be. So we can have muscular contractions, very much like peristalsis, because of that thick muscularis. Plus we have an epithelium that's ciliated to help move these structures where they need to be. Excuse me. Right down here, we just have a cross section through the testes or through the epididymis itself, and you can see all the convolutions of what will eventually form the ductus deferens. So, just going through in words, structures that we've talked about several times now, right? Immature sperm are going to basically leave the epididymis, or actually they're mature by the time they're in the epididymis, so I misspoke. Mature sperm mature in the epididymis, right? When called upon, they're going to be transported down the ductus deferens, also known as the vas deferens, right? They will travel up the spermatic cord. They're going to reach the ampulla. From the ampulla, that material, that sperm, is going to then mix with the product of the seminal vesicle, right? Then it enters the ejaculatory duct, which is in the prostate. The prostate then adds more material, and then that material eventually will be expelled from the body. But the ejaculatory duct is formed by the union of the ductus deferens and the duct of the seminal vesicle. So it's found inside the prostate gland, and it's about a centimeter and a half long. So that leads us to the urethra. And if you remember from our section on the urinary system, the male urethra is subdivided into 3 distinct regions. Prostatic urethra as it moves through the prostate gland, membranous urethra as it passes through the muscles of the pelvic floor, and then the spongy urethra, so named because it's near the corpora spongiosum, or spongy body. So let's talk a little bit about these 3 glands, which are necessary for the production of semen. So the 3 glands, So the function of this material is to help nurture the sperm. They're going to need protection from the harsh environment inside the vagina. They're going to need an energy source. So one of the things that you're going to find is overall semen tends to have a basic pH. It's going to be slightly caustic. This is a way of neutralizing the acid environment of the vagina and making it a little bit easier for the sperm to survive the trip, right, all the way up the female female reproductive tract, so we can get to the ampulla of the uterine tube, right? That's, for a single cell, that is a long journey because they have to get all the way up into the uterine tubes where fertilization normally takes place. Okay, so we'll talk about each of these glands in turn and talk about what they're actually going to be contributing. So the seminal vesicles, these guys right here, seminal vesicles, right? Seminal vesicles on the posterior side of the bladder, right? They're going to secrete a viscous, whitish yellow alkaline fluid containing fructose and prostaglandins. Okay, let's break this down. Alkaline, because this is what's going to give semen a basic pH, again, to help protect the sperm from the acid environment of the vagina, right? They'll be containing fructose, right? A sugar. So this is an energy source for the sperm. They got a long way to travel. They're going to need a source of energy to get there. Here's where it's coming from. And the last one, prostaglandins. Prostaglandins are short-range chemical signaling molecules, right? They can do lots of things. It's ways cells talk to each other. In this particular instance, the reason that prostaglandins are being added to semen is because this will lead to the relaxation of smooth muscle. So if you can imagine our reproductive systems coming together, right, semen are going to be deposited right outside of the cervix. Remember, the cervix is that enlarged entrance to the uterus, right, at the large entrance to the uterus. This is made of smooth muscle. So when the prostaglandins made by the seminal vesicle come in contact with the cervix, it causes that smooth muscle to relax and opens that external os of the cervix, making it possible, or at least easier, for the sperm to gain access to the uterus. Then they can swim up the uterus to one or the other of the uterine tubes. So prostaglandins are a chemical way for sperm to sort of politely knock at the door and so here they are located behind the bladder, right next to the ampulla. So the next structure here is the prostate gland itself. Here you can take a look at the histology of it. The prostate gland is immediately inferior to the urinary bladder, right? Its materials are directly secreted into the prostatic urethra. So this material is added basically during ejaculation or immediately preceding that. Prostatic secretions are slightly acidic, we'll get to that in a second, contain mucin, so when this mixes with water, it's going to be mucus, citric acid, which is a nutrient source for the sperm, but it explains why this particular component is acidic. However, semen overall is basic, but this particular one is a little bit acidic because of the citric acid, which is a nutrient for the sperm. Also, there is a compound called seminal plasmin. Seminal plasmin is an antibacterial compound, right? The vagina is kind of a hostile environment to sperm. There are bacteria in there that would be perfectly happy eating that sperm. So they have a chemical protection versus bacteria in the vagina. Also, you're going to have a compound called prostatic specific antigen, PSA. Right, so this is an enzyme that helps liquefy semen. So this makes it easier to deliver semen. Otherwise, it would be, the more viscous it is, the more difficult it is to transport. So what PSA is, what its function is to liquefy semen to make it easier to transport. Now, what PSA is used for medically is an indication of prostate cancer. So those of you who have talked about or heard about people getting their PSA checked, or they're suspicious of having prostate cancer, this is what they're looking for: prostate specific antigen. You can run a blood test, check the level of this antigen in your blood, and it may be an indication of prostate cancer. Now, about 10 or 15 years ago, people took this as like the critical sign. If your PSA goes up, we have a blood test, you have prostate cancer, and boom, you're going to get surgery. So what they found, or what they started doing, is if you had really high PSA levels, which they thought was one-to-one always connected with the severity of cancer, if your PSA levels were high, they would just pull out your prostate. Right, boom, it's just gone. They're going to surgically resect it. Unfortunately, there are consequences to that. Very common consequence of this would be urinary incontinence. You can't no longer control when you go to the bathroom. Impotence was certainly, or the inability to have an erection, very common side effects of the surgery. And over the long term, they found it didn't necessarily really cut down on the severity of cancer cases in these individuals. So they were taking this very aggressive surgical approach, but over the long term, it turned out not to really be worth it. So nowadays they take a much more nuanced approach into checking your PSA levels and see whether or not you should have surgery. So now still checking your PSA levels is an important part of checking men's health. If you have elevated PSA levels, it's considered an indication you might be at greater risk. So what they do now is, instead of just ripping the thing out without thinking about it, they're going to start doing more frequent biopsies and just taking a lot closer look before actually taking any surgical action, because it really wasn't doing all that good and was perhaps doing more damage. So you'll see a lot of kind of conflicting information out there. PSA is still a good thing to check, but just realize it isn't the whole story, right? It's a good indication. It's an easy test to do to see if you need further testing. Then when you get your biopsy, they can have a much better idea of what's going on. Oh, one other thing I should point out, right? The next most common, or probably the most common way of checking the prostate as an indication of prostate cancer is something called the DRE, digital rectal exam. So what happens is the doctor is going to insert his or her finger into the anus, the rectal canal, and they're going to palpate. They're going to touch your prostate gland. So the reason why this is done is to see if the prostate is swollen, is it abnormally large, does it have any lumps in it, you know, is it basically, it's like doing an exam to check for cancer, right? Do you have any hard, strange masses in there? Is this organ swollen up? And this is a test that starts to be performed on men around 40 to 50 years of age, depending on family history and stuff like that. So it may not necessarily be enjoyable for everyone who goes through it, but it's a lot better than getting cancer. So encourage dads, uncles, brothers, whatever in your life. They may be a little bit squeamish about this, but it's certainly a lot better than getting cancer. So that's the way I think most people should handle that conversation. We'll go back to where we left off. Oh, one other thing I want to talk about prostate cancer. It has now come to be fairly clear that most people, most men later in life will develop prostate cancer eventually. I think I saw a statistic somewhere, by the time that they're 80 years of age, most men, like the majority of men, have some low detectable level of prostate cancer. It just seems to be a thing that always happens. So reacting very strongly to these signs isn't particularly helpful. Yes, your average 80-year-old is starting to develop prostate cancer. However, it might take 30 or 40 years for that cancer to kill the individual, but they're probably not going to live to be 130, so something else is going to get you first. So it's really not that big of a deal, but it just is a way of explaining kind of a complicated situation where a lot of men, when they get older, they're going to have signs of this, but you need to ask sort of very nuanced, specific questions like what kind of prostate cancer is, how aggressive it is, how fast is it spreading. This means you need to keep a close eye on it. Keep measuring the PSA levels, keep getting those DRE tests. If you need biopsies, go ahead and get those, because something that looks like a problem may not actually be a problem, but if you don't keep an eye on it, it might get away from you. So just a little bit of health facts there. Next set of glands are the bulbourethral glands, right? These are on either side of the urogenital diaphragm, in that right next to the membranous urethra, right? The job of these glands is to produce a mucus solution that just basically helps serve lubrication and basically getting your urethra ready to deliver semen. So, right, the seminal fluid made of these three accessory glands combined with sperm from the testes, or it's going to make up semen. So the process of releasing semen is called ejaculation. On average, there are about 3 to 5 mils per ejaculation, containing nowadays around 200 to 250 million sperm. This number has been dropping actually for decades. Most of the people, most of the men in this class, compared to your grandfather, you have probably a half to a quarter of the sperm count of your average grandfather. So sperm counts have been dropping substantially over the last few decades, and it's not really understood why. It has been hypothesized that it may have to do with a less active lifestyle or having a greater number of hormones particularly in like meat, right? A lot of hormones are basically injected into animals, and that gets into our food. It also gets into the wider environment, which may be affecting sperm counts. The good news is 200 million sperm is like way more than enough. So it's usually not that big of a deal, but it is something that people have noticed, right? Sperm counts are dropping, right? We don't entirely know why that is. It might end up being more of a problem in the future. Those people who are already struggling, trying to conceive, this certainly isn't helping whatsoever. Other potential causes for this would be increased environmental toxins. So basically, the more pollution you have in the environment, the more likely that's going to get into the system, the more likely that is going to disrupt the delicate process of sperm production. So you're going to lose more sperm in the process of trying to create it, right? So if you start with 100 million, but half of them get messed up because of environmental toxins or developmental problems, right? You start with smaller and smaller and smaller. So it could be lifestyle changes, lack of exercise, being overweight, environmental toxins, increased levels of stress. There are lots of reasons put forth to try to explain why sperm count has been dropping substantially in really across the world for several decades now. So let's talk about copulatory organ, which is the penis, right? So the penis and the scrotum make up the external genitals in men. We have several regions of the penis. We have the root of the penis, which is actually located inside of the body. It is made up of the bulb and the cura of the penis. Let's just go to the picture to make that clear. So here are it's our pubic bone, right? Here is what's called the crus of the penis, just like we had a crus of the clitoris. Same thing, same embryologically derived erectile tissue anchored in the exact same location, that inferior pubic ramus, right? So right crus of the penis, we have another erectile ball on the other side, the left crus of the penis. These two are going to come together and form these two regions here. So these two regions here are the corpora cavernosa, the cavernous bodies. Spongy-like erectile tissue, fills with blood, right? Veins close off, so the whole tissue just swells up and increases in size. Down here is the third erectile body called the corpora spongiosum or corpus spongiosum. This is the one that has the urethra passing through it. Because that's the spongy body, that's why that is the spongy urethra. Now, the corpora cavernosa, right, they start over here at the crus of the penis. The corpora spongiosa starts over here, and that forms the bulb of the penis back here. And you can see the membranous urethra entering the bulb of the penis here. Right, so that explains that, right? So the body of the penis, or just the shaft of the penis, is made up of these three erectile tissues, right? Two corpora cavernosa, one corpora spongiosa, surrounded by a layer of connective tissue called tunica albuginea, right? So another layer of connective tissue which provides structural support to these erectile tissues. If you get sufficient trauma, you can actually rupture the tunica albuginea and the penis, and you have a condition called penile fracture, where you're going to get almost like a balloon growing out of the side of the penis, because that is where the tunica albuginea has ruptured, and then that erectile tissue will start protruding from that. Okay, so the very end of the penis is called the glans, which will contain the external urethral orifice. So I do believe we've already covered most of these structures. Pardon me from jumping back and forth. This particular image does not show the prepuce. It does not show the foreskin of the penis. right? This is sometimes removed in particular communities called, with a surgery called a circumcision. There is some medical debate going on whether or not male circumcision is really warranted. It appears that in the developed world, at any rate, it doesn't really appear to provide much protection or better the health of an individual. It is certainly done by various cultures across the world. I'm not going to comment on that, but the original medical reason for doing this was it was thought that would lessen the likelihood of contracting or passing on sexually transmitted diseases. And for those people who have challenges with hygiene, it made it much less likely that you would pick up a skin infection. So it became common medical practice that for health reasons, you would always perform a circumcision on men. More recent studies have shown that this is probably not necessarily the case. Certainly people who have access to good hygiene, there's no real strong evidence to support this claim that it's really health related. To my knowledge, there was only one study done about 10 years ago that showed in one particular population, in one particular part of the world, that circumcision would reduce the likelihood actually of HIV infection by about 10%. But that's a very special case of one particular disease in one particular part of the world that was done some time ago. So what you're seeing is the rates of circumcision, certainly in the United States and in many other parts of the world, starting to drop because there isn't really strong medical evidence to back it up. But the sort of cultural reasons for doing it are completely separate, and I'm not talking about that because this is an anatomy class, not a anthropology class. Okay. I promised you guys that I would talk about some alteration to male anatomy, right? We talked about the presence, or at least in humans, right? In females, breasts are pretty much constant after puberty, which is unlike most other mammals. And we had hidden ovulation in human females, which is different than most other mammals, right? There have also been some pretty big changes here. What is missing from this picture? Right, take a look at it. Something important is missing. Right, think of every major projection off the body. What do you have in there? Right, bone. There's no bone here. Right, even your nose has a bone in it. Very minor projections off the body always have bone support. This one does not. And originally, there was a bone. There was actually a penis bone in the ancestors of humans. Penis bones are a very common thing in the animal world. They're called baculum. So the baculum in human beings, and actually in our closest relatives, is either absent or it is vestigial to the point of almost being gone. So if you look at gorillas and chimpanzees, they do have a baculum, but it's so teeny tiny you can barely actually find it. It doesn't have much of a functional role. Whereas you look at other animals, dogs, raccoons, or something like that, you will find a very prominent baculum, right? A penis bone. And if you think about it, that makes total sense, right? If like your ability to pass on your genes is dependent on the function of this organ, wouldn't it make sense to generate an erection instantaneously and have it for as long as you would possibly want, right? That would make it a lot more effective to pass on your genes. But this has been lost, right? This has been lost from the male anatomy. And again, the reasoning is, the belief is to heighten or to encourage, evolutionary speaking, pair bonding. Just like with hidden estrus, right? If you don't know when the female is ovulating, it means you got to stick around and have a whole lot of sex. You have to stay around and be around all the time throughout the entire month, and basically be mating that entire month in order to basically have a chance at fertilizing an egg, right? Same evolutionary pressure here, right? Not only does basically it ensures pair bonding, right? You have to have an emotional investment in the individual in order to be, right, excited enough to activate this erectile tissue to have intercourse. Also, it's pressure from the female on the male, right? In order for this to work, you have to have a strong cardiovascular system. You have to be in very good health, right? If you're not in very good health, this is not going to work, right? If this is not going to work, you're not going to pass on your genes. So this whole sort of alteration in human physiology coming to the reproductive system, and by the way, I should put forward, this is not like 100% accepted, but it's a fairly well accepted theory. Lots of people start arguing about it, so don't take it as fact, but it is a at least internally consistent explanation for why the human reproductive system is different than in many other animals, particularly in many other mammals. So the idea is, right, if you have two individuals together working in team, you have a much higher likelihood of taking a newborn child and bringing it to adulthood where it can be self-sufficient, right? Raising a human child is really, really, really difficult. Anyone who's ever done it or seen the process, it's hard, right? Human beings are pretty much, babies are worthless, right? They just sit there and they demand to be fed and they poop themselves. That's all they do. Think about another ridiculous animal. Think about a giraffe, right? There's an animal that's all kind of weird and messed up, right? You watch a program on the Discovery Channel, pregnant giraffe is walking around, right? Gives birth, giraffe drops like 6-7 feet, like the baby giraffe drops 6-7 feet. 5 minutes of things like standing, right? 10 minutes after that, it starts to walk. 15 minutes after that, it's running, right? It's not even, it's like an hour old and it's already running. Right? It takes a human child like a year and a half to be able to do that, right? And the reason is this enormously huge brain, it's got to fit out that really tiny pelvis. So human children are born about 3 or 4 months early. If you compare human beings to other animals with a similar size, we should be pregnant. Human beings should be pregnant 12 to 14 months. However, if they stayed in that long, the cranium would get so big, the brain would be so big that childbirth would be impossible. So they have to come out early because of our giant brain. Because they come out early, it takes a lot of work to take care of them. So there are all kinds of evolutionary pressures that have been put on human beings to try to get humans to work together to raise an individual child. And this whole theory goes on to explain lots of other things, like why do children never sleep very well, right? Why do newborns want to feed every 3 to 5 hours? And the theory goes that it's actually deliberately to prevent the parents from having sex, right? If the child's waking up every 3 hours, you're going to be tired and exhausted. If you're tired and exhausted, you're not having sex. If you're not having sex, child number 1 does not have to compete with child number 2, right? So if there's a second pregnancy, that is a major toll on the physiology of the mother, right? Mother still needs to nurse baby 1, but if it gets pregnant with fetus 2, that's a huge draw on the physiology, and that actually threatens the health and life of the first child. So I think one of the reasons that kids have the sleeping cycles they do is to impose space between the children to make each birth or each pregnancy that much more likely to actually reach full term. So could go on for hours and hours and hours about all this kind of stuff, but I know you guys just want to be finished with this class, so I will stop this story here. Again, I just want to say this is not like conclusively accepted facts, but it's a theory that is moving forward, that is gaining more followers. So if you're interested to know more about that, there are all sorts of classes and YouTube videos you can check out about that. Maybe I'll post some at the end of the section. Let's finish up these slides. Okay, so as we mentioned, the shaft of the penis contains 3 erectile bodies, 2 corpora cavernosa, which are on the lateral sides of the penis. The corpora spongiosum, which carries the urethra, is on the inferior surface, right? And will actually widen and form the glands of the penis. So these erectile bodies are also known as venous spaces because they fill with blood, and that's how you generate an erection. So maybe be of no surprise, the parasympathetic nervous system is responsible for erection, right? So this is your rest and digest, controlling blood flow into the penis. So erection is controlled by the parasympathetic division of the autonomic nervous system, while ejaculation is controlled by the sympathetic division of the autonomic nervous system. This is your fight or flight response, right? And timing these two out can be important, because if you have activation of your sympathetic nervous system before your parasympathetic nervous system, right, you're going to have premature ejaculation. Okay, so if I haven't already explicitly stated, ejaculation is a process by which semen is ejected out of the man's body. That's a really weird and sort of creepy thing to end the semester on, so I'm just going to show you a cartoon picture of a developing fetus, because that is what this entire section is really all about: human reproductive system. So remember, the reproductive system is the only body system that the individual, the only body system that you can live entirely without. Take away any one of the other body systems, you're going to die, like, immediately. However, reproductive system, you can live without. You can live a perfectly full life without this. From the evolutionary point of view, all of those other body systems exist so you can have a reproductive system and pass on your genes and keep passing on that self-replicating DNA molecule from one generation to another, to another, to another, to another. So that wraps up the content of this class. I will probably post a short video kind of wrapping up this whole class, giving you guys some parting thoughts, but I will post that a little while later. Till next time, take care.